Hi everyone, I'm back. This is Kevin. I'll be your moderator for the Q&A. With me on the panel, I have Mr. Danny Ong, Dr. Goh, Dr. Thornbeck, Dr. Zhou, and with me on the call, I have Dr. Yong, Dr. Fong, and also Prof. Slack. Yeah. So I'll be moderating some of the Q&A that has been posted right here on the Q&A chat. So I think the most voted for question is that the recently circular RNA is gaining traction in research. So do you also see this in liquid biopsy? And is there any connection to microRNA? So maybe I can pose this to Dr. Zhou, Dr. Frank, and also Dr. Thornbeck. Yeah, so Dr. Zhou first, maybe go ahead.
I think Frank is probably the best to start that.
Yeah, so yeah, Prof. Slack.
Frank, we can't hear you.
I think you're on mute.
Yeah, maybe we move on to the next question first while we sort out some of the technical issues. Yeah, so the next one is how sensitive is PCR-based assay in detecting single nucleotide change in miRNA? Yeah. So Dr. Zhou?
Sure, I'll take that. I think for microRNA or any nucleic acid, there's always different methods. So namely sequencing microarray and PCR. And I wouldn't say any one particular method is better than the other. It really depends on the context of application. The reason why PCR, when we look at the clinical application of microRNA, it has been almost 100% using PCR because these microRNAs are extremely small. So to optimize an NGS or microarray-based microRNA test has been extremely difficult, and also the affordability of running those for a small panel of microRNAs are probably not as economical as PCR. So when it comes down to single nucleotide differences or differentiation of those homologous family members, it is one of the key analytical validation step when we launch any microRNA test. So in the 12 microRNA GastroClear, beyond the clinical validation, we actually conducted extensive validation of its closest family member. And generally speaking, we are able to achieve at least a 50-fold differentiation of single nucleotide differences. But again, we have to put that in the context of the clinical problem. So if we have two very similar microRNA, which differ by 1,000-fold in the clinical sample, then a 50-fold analytical specificity is insufficient. But fortunately, when we look at many of the circulating microRNAs, the biological differences is less than that, which means a 50-fold analytical specificity is more than sufficient, at least for all the disease we have seen so far.
Thank you, Dr. Zhou. All right, I think we are back online with Prof. Slack. So maybe we'd like to go back to the first question. Prof. Slack, I'll repeat it once more. So recently, circular RNA is gaining traction in research. Do you also see these in liquid biopsy, and is there any connection to microRNA? Prof. Slack, please.
Yeah, sure. Can you hear me now?
Yeah, we can hear you.
Okay, great. Sorry about that. I had to restart the webinar. Yes, so you're, as I was saying earlier, the people asking the questions are clearly following the latest trends in RNA research, and circular RNAs are indeed emerging as important players in the RNA networks within cells. So circular RNAs are the result of a backsplicing event between sort of introns and exons, and can be detected by a number of different approaches, including like a PCR-based approach, which amplifies across the junction that will be formed in the circle. They are very new, and so the work on them as biomarkers is just beginning. I think I mentioned in my talk that there were over 900 clinical trials with microRNAs, but there are only 2 at the moment with circular RNAs. But as I've spoken to Li Han and the Merix's team about in the past, I think that this is going to be a useful area for them to try and develop tests around, especially for brain disease. I mean, these circular RNAs are found very extensively in the brain.
Yeah, thank you, Prof. Slack. Right, next we have a question for Dr. Goh. Right, so how do clinical labs minimize pre-analytical variations, and what is the minimum number of samples recommended for validation? Dr. Goh, please.
Okay, on the first question first, what we did is to get the phlebotomies to indicate the time of blood draw on the blood tubes, so that when they are sent to the laboratory, we ensure that it's processed within the required time frame. In this case, it's within 2 hours, right? And after the spin, we will also check for hemolysis. I think Li Han showed a color chart earlier. So if it's like quite orangey and quite red, then that will indicate hemolysis, and the sample will be rejected. On your second question, the standard doesn't specify exactly how many samples are needed for validation. So I think it depends on the complexity of the assay as well as the number of analytes to be analyzed. In this case, because it's new, so we decided to validate 50 as a minimum specimen requirement. Most of the time, our minimum requirement is 20 samples, but in this case, we validated more.
Thank you, Dr. Goh. That was very insightful. Next, we have a question specifically for Dr. Yong. So you mentioned that there was new ground broken with the creation of SS656. So with regards to standards and its roles in innovation, is it always a positive impact, or would there be instances where there have been adverse and inverse impacts? Dr. Yong, please.
Okay, so that's I would say it's a good question. And answer is a bit better. So what I mean is, look at standards, there are various types. For example, you have manufacturing standards, which is to do design and development purpose. And also, I think recently become innovation, a lot of it is related to operating standards or adopting. So in our experience, especially we actually have seen spectrum standards created for different stages of maturity of either or a operating model of design. So to me, in terms of if you're asking what helps, especially from a new process and perspective, it's always the more from the passion operating. Right? So let's say it's very research stage do with going with policy. So generally it's positive because I think standards help to actually introduce the area in wider industry. But at the same time, I would say you have a time when you create standards because early you might exclude some other approaches to solving the problem. Not relating, let's say, case, but more of, let's say, the remodel process. So that is the time coming from experience where I sit in the perspective, looking at development of standards in regard to innovation, research and development.
All right, thank you, Dr. Yong. Next, we have a regulatory related question from Mr. Danny Ong. So you mentioned about product registration. So if my RNA assay lab is a lab developed test, do we also need to submit it for product registration?
Okay. So the lab developed tests, by definition, they are diagnostic assays that are developed within the lab, manufactured within the lab, and then for use only within that particular lab. So we have scoped the SS 656 standard to include both in vitro diagnostics, medical devices, as well as lab developed tests or LDTs. But in terms of product registration, the LDTs are actually exempted from product registration, but they are more managed under the PHMC lab license requirement. Yeah, so actually product registration is not required for lab developed tests.
Thank you, Mr. Danny Ong. Right. Next, I'd like to just put a question on the floor for anyone that wants to answer. So, do you see the need to develop mRNA diagnostic to replace the current tumor markers such as CA125? If yes, what is the gap to fill now to move that forward?
I think that things like CA125 and actually, funnily enough, many years ago, I was one of the team that helped to develop the CA125 years ago. That they are very old markers and they're very non-specific markers. I think we've made a lot of our knowledge of tumor biology, our knowledge of what we're trying to, the patient populations and how diagnostics are used has changed dramatically. We saw on the slides I put up that they can be predictive, prognostic, or diagnostic tests. And I think that what we're seeing with the microRNA based diagnostics is we have a much wider possibility for developing specific tests, which is specific targeted populations for specific indications for use, which I think that you don't see with simple protein biomarkers.
Right. Thank you, Dr. Thornbeck. Dr. Zhou would like to add on something as well.
Yeah, if I may add on, as much as we are microRNA centric, I think the future of medicine is multi-analyte tests. I would say protein tests has been most widely implemented. Even today, probably 90% of the routine clinical tests are done on proteins. Right? So I was still advocate that there are a value to protein tests, but as John has mentioned, there are limitations. So to me, I believe in the future, the physician will look at a combined report of DNA, mRNA, microRNA, and proteins. And depending on the disease application, maybe one of the classes will outweigh the other. But so what we are advocating as a developer is always a plus approach instead of replacing. I think in the clinical world, if we can manage the workflow to provide the best information to the clinicians at the most affordable price, combining multi-analyte is actually worth exploring. And of course, we see distinct advantage of each of the class of biomarkers, and particularly in the case of CA125, there is an ongoing project we have with NUH, DTSH, and DXD, specifically to look at the combinatorial power of microRNA plus CA125, which really give us 1 + 1 more than 2 effect. So my take is we try to combine to give more clinical utility, but the process and the cost must be managed.
Right. Thank you, Dr. Zhou, and also Dr. Thornbeck. So to tag on to your point on looking towards the future, I have one for Prof. Slack. So how do you see microRNA diagnostics impacting personalized medicine? Prof. Slack, please.
Yes, this is clearly, just to add on to what Dr. Zhao was saying earlier about how medicine is sort of moving to multi-analyte tests and how a physician is going to be looking at both genomics and transcriptomics and proteomics in order to make their decisions. We see microRNAs as sort of one tool in that ongoing sort of march towards personalized medicine. We've looked at the prospect of combining genome sequencing with microRNA detection as a potential better way to sort of decide which patients are going to respond to certain therapies. I mean, that's the sort of ultimate goal precision medicine that we'll be able to tell physicians exactly which drugs each patient is going to best respond to. And one thing that I would just point out is that we've talked a lot about RNA today as being a diagnostic marker, but in fact, one of the arms of precision of sort of RNA medicine is in fact being able to either deliver RNA as a drug or target RNA as a drug. You already know about some of the drugs that are approved, like siRNA drugs, these RNAi drugs that will target a messenger RNA as a therapeutic option in certain disease. We also have examples of delivering RNA. An example that's quite popular right now is to try and devise RNA-based vaccines against, for example, SARS-CoV-2. So at some point, we're going to be able to combine the genomics, the transcriptomics, to not only decide what current therapies might be available to patients, but also maybe try and design an RNA-based therapy that would be appropriate for that patient as well. Thanks.
Right. Thank you, Prof. Slack. So on synergies, I have a question here for Dr. Fong. So Dr. Fong, what are the pros and cons for mRNA as a biomarker compared to, let's say, maybe DNA and protein? And also, are there any synergies between the different types of biomarkers? Dr. Fong, please.
Yes, microRNA, can you hear me? It has an advantage of being small and overall much better lipid profile. I think that's one of the major. And also integrated in many more numbers. Otherwise, not advantage. I think the challenges we have Professor Slack as indicated that very small size microRNA observation and accurate data. And in terms of combination, Neha already mentioned as well, Professor Slack. I think in the future, multi-variant, potential biomarker type is set will actually provide much better specific, especially for heterogeneous diseases. I'm sorry, I'm hearing a lot of echoes, so that's been difficult for me to speak. Did I answer the question?
Yes, thank you, Dr. Fong. All right, so I have one specifically for Dr. Zhou. We know that you mentioned that you have learned a lot of lessons throughout your whole journey developing GastroClear, and SS656 is sort of a summary of your lessons learned and also helping people overcome such difficulties that you have come across, the challenges and difficulties. So what are the key lessons learned from developing and deploying GastroClear?
Right. Yeah, so to take it short, I mean, GastroClear took us 6 years from doing the first experiment on the bench to getting the dossier ready to submit to HSA Danny's team. I think it could have been done a lot faster, but as we mentioned, the pre-analytics part was something we didn't know. So we got into discovery, and as Danny's last slide has shown, we basically went into product development without resolving some of the pre-analytical steps, which essentially means we had to re-kickstart the whole process. So that took a lot of iterations, and I think that can be avoided from the moment that clinical enrollment start to either get the patient samples, biomarker discovery or validation. That's extremely important, as Danny has said. If we didn't start with the end in mind, and if we didn't know all the processes in between, chances are we're going to fail at one of the steps. And for medical device, there's really no room for failure, right? So I think pre-analytical, and during the analytical steps, as Ling has mentioned, there are 4 key steps within the lab itself, right? How do we design a product which can be widely applied, not only in the research lab where it is discovered, where the lab had the skill sets, the knowledge, and probably the equipment, which many of the clinical lab probably do not have. So again, the usability of the device is also something that I think all developers need to think about in order for wide clinical adoption. And the third thing is really, how do we then present the data to a busy clinician who doesn't have all the time to read microRNA papers or to look at what's the latest in circular RNA or any other research setting, right? So the interpretation of that medical device and the actionability of the medical device is also something extremely key. Otherwise, we are developing a research tool that will remain on the bench and never reach the bedside. So I think the core lesson learned in GastroClear is really making sure that we have the end in mind. We have probably think through all the processes with the goal of engaging the clinician to implement the test. So that's some of the things that we recommended to SS656.
In my experience, it's I'd agree with what Dr. Zhu said, but in addition to talking to the clinicians, you have to actually be involved in what the laboratory does as well. One of the in other companies which I've run, one of the big issues has been, although the clinicians might want to order the test, the lab will go, well, actually, we don't have the technicians trained. It's not enough tests for us to bother to do that, or we don't have the right equipment, or we just don't get those requests at the moment. So we're not going to run the test. So you have to think when you're developing a product, not only the clinical need, but actually the laboratory need, the laboratory. So you have to involve both the laboratory specialists and the clinicians. The clinicians are a pull, but also you have to really get the laboratory scientists on board as well. Otherwise, you just get blocked. They'll say, well, actually, we just don't have time. We're laboratories, are always understaffed and overstretched. Clinical laboratories. So you've got to try and make it as easy as possible for them to make that decision to offer that test. And I think that's a very big lesson learned.
Thank you, Dr. Zhou and Dr. Thornbeck. So I think to tag on the lessons learned from 656, on the lessons learned from the development of GastroClear, how would SS656 then help to standardize and accelerate implementation of mRNA diagnostic tests?
Right. Maybe I can start here. I think SS656, I'm hoping that it will be read not only by the industry groups or the clinical labs or the regulators, but in fact, I think some of the thinking process behind it should be made available to any translational researchers who are hoping to bring a new discovery for research to a potential bedside application. Right, so that includes many of the pre-analytical and the clinical utility thinking. And what Yulian has shown, we typically have tens of thousands of biomarker publications, and only 0.1% of those turns into a clinical product. And the reason is not because the research labs are not doing their job, but they were not informed of the many aspects that I think we have talked in all these presentations to bring something into the clinical setting, right? So I'm very much hoping that SS656 is not just an industry standard, but something that will guide people to think about microRNA or any RNA test development.
Thank you, Dr. Zhou. So I'd like to put one on the floor for anyone to answer. So what are the steps considered to improve sensitivity and specificity before deeming the mRNA is not suitable as a biomarker and move on to the next target? So maybe this one is more for Dr. Thornbeck, yeah.
That's a tough one. When you're doing the biomarker discovery, I should have brought the team along, shouldn't I? To answer this question for me. Dominic, where are you? Because we talked about this the other day, actually. And I think what you're looking for is when you're looking at biomarker discovery, one of the things you have to really have is very well curated clinical samples. That's the big thing. So you've got to have, if you're looking to differentiate between a patient population and a normal population, you have to be very sure that you have those patient samples very well curated, and that's the essence of it in general, because that allows you then to identify the differentiation between the clinical situation and the normal, so-called normal patients. And if you don't have that, then you'll never see the differences, which is especially in something like the microRNA, where you've got it's going to be a multi-analyte. It's not going to be, you're not going to pick up one and pretty well, never going to pick up one microRNA that's going to be specific. It's going to be a mixture like GastroClears 12, and then you're having an algorithm to do a risk analysis. So it's all about having well curated samples, clinical samples at all parts of your process that allows you to differentiate. That's the key.
Thank you, Dr. Thornbeck. We have one here specifically for ProfSLAC. So Dr. Yong mentioned that variability between findings of research groups is known. And in the development of this standard by DXD Hub, could you summarize the steps taken to validate the accuracy of the standard?
I'm not too sure if I'm the best person to answer that. Probably Dr. Zhao is better suited to answer that, because we haven't as yet tried to validate this particular standard. I can tell you though that we use the MerXS technology in our lab, and we use the protocols. But you know, we've basically followed the protocols as opposed to sort of try to, you know, improve upon them or validate them at this point.
Thank you, ProfSLAC. So I'll just round this up with maybe the last few questions. So since the future of diagnostics are multi-omics, how should we prepare ourselves from data overload, and where we draw the line to say the product is now good enough to push for commercialization? Maybe Dr. Thornbeck or Dr. Zhou?
Well, the key question is what's going to be the clinical utility. That's always got to be your question. You know, the doctor doesn't care what the technology is; they care about the result. So, if you're not bringing more information that allows the clinician to make a better clinical judgment on how to treat that patient, then you don't have a diagnostic. That's the bottom line. Right, so you work until you've always got to be focused on the clinical utility. That's the message.
Thank you, Dr. Thornbeck. So I think we'll have our last question here. So this is for ProfSLAC again, and also looking into the future again. What is the next frontier of microRNA research and clinical translation?
Wow. I feel personally that it's going to be a combination of microRNA diagnostics and microRNA therapeutics. So in some cases, there are sort of well-documented examples, like the example that was mentioned earlier about miR-21, that looks like it's upregulated in many different cancer types, and it's clearly an oncogene in many different cancer types. And so you could imagine a diagnostic to examine the levels of miR-21 followed by a therapeutic that targets miR-21 to sort of knock down the levels. So you have sort of a theranostic approach here where the RNA is both the companion diagnostic and the therapeutic target. But, you know, that is, you know, I mean, while there are clinical trials already targeting microRNAs in a variety of cancers, it's not in widespread application right now. So this is something sort of in the future. I think another point that was brought up is this idea of using microRNAs to understand the actual disease. So not only knowing which microRNAs get secreted into the bloodstream, but also looking at the tissues themselves and asking, you know, what are the misregulated RNAs? Because as was shown, that can help you build pathways and so help you understand the disease and identify nodes that might be targetable in that particular disease. So I think there's a lot of low-hanging fruit. There's a lot of wide open questions. Certainly, you know, there's a lot of work that needs to be done. I mean, you know, John mentioned that there were 600 microRNAs that were easily detectable in human serum, but there are about 2,500 of them that are made in our cells, and there are probably 250,000 variants of those microRNAs. So really, just trying to understand what these RNAs are doing and how they're impacting cellular processes and health is going to keep us busy for a very long time.
So unfortunately, we run out of time for our Q&A. Sorry we cannot answer all of the questions. So here I'd like to thank my presenters and also panelists for joining us today, and also everyone who's viewing this online. Thank you for joining us, taking time out to join us, and have a great weekend ahead. Thank you.